13-week dermal toxicity study of VIM-004 gel, a star particles-based formulation in Göttingen Minipigs. Abstract. Vitiligo is a skin disorder that affects melanocytes, leading to depigmented patches on the skin and mucosa. Studies have indicated that melanocortin system defects, notably decreased level of alpha melanocyte stimulating hormone, alpha MSH, contribute to the disease mechanism. Despite various therapeutics options targeting melanocytes, treating vitiligo remains challenging due to the complexity of achieving safe and effective drug delivery to get the desired degree of repigmentation in the lesioned skin areas. This study explores VIM-004 gel, an innovative gel formulation containing an alpha MSH analog, alpha melanocyte stimulating hormone analog, VIM-004, combined with a star-shaped particles. The star particles in the gel, when administered with the VIM gel by mechanical rubbing on the skin, create microscopic pores through the stratum corneum. This approach temporarily increases skin permeability and allows for delivery of the therapeutic agent VIM-004 in the epidermis and dermis layers to produce effective vitiligo treatment and reduce systemic toxicity. This study evaluated the potential local and systemic toxicity of VIM-004 gel administered to Göttingen Minipigs by topical administration once daily for 90 days. In addition, the toxicokinetics characteristics of VIM-004 were also determined. 9 Göttingen minipigs were randomly assigned to 3 groups: placebo, low, and high dose. The placebo and test article were applied daily for 90 days to cover at least 5% of the body surface area, BSA. The following endpoints and parameters were evaluated: mortality, clinical observations, including dose site reactions, as well as local skin indications of irritation, erythema, and edema, food consumption, body weight, transepidermal water loss, TEWL measurements, skin puncture, ophthalmology, electrocardiography, and clinical pathology parameters. Blood and skin biopsies were collected at multiple time points to characterize VIM-004 concentrations, both systemic and local, respectively, over time. If you want more information about my poster, I'll be happy to meet you at my poster presentation session to take any question. Thank you very much for visiting my poster.